Hello everyone. In this video we will discuss about the two most important tumor suppressor gene that we need to know. First is RB gene or retinoblastoma gene, located on the chromosome 13q14, long arm of chromosome 13, and this inhibit the cell cycle progression at the G1S checkpoint of cell cycle. So normally growth inhibitors like TGF beta, P53 and others, they stimulate the cyclin dependent kinase inhibitors like P16 and INK4A, that will result into the inactivation of cyclin D, CDK4, cyclin dependent kinase 6, cyclin E and CDK2. So this will result in hypophosphorylated RB gene. Hypophosphorylated RB gene is an active form of RB gene. This hypophosphorylated RB gene bind the elongation factor E2F, which is a transcription factor, and will result into the transcriptional block and no cell cycle progression beyond the G1 point. On the other hand, growth factors like AGF, PDGF, they activate cyclin D, CDK4 and 6, cyclin E and CDK2. They will result into the hyperphosphorylation of the RB gene and make this gene inactive. Hyperphosphorylated RB gene, this is RB gene. Hyperphosphorylated RB gene release the E2F elongation factor and will result into the transcriptional activation and progression of cell cycle. So this is the normal functioning of RB gene, and the various factors like TGF beta, P53, CDK inhibitors and their role in its activation and inactivation. Retinoblastoma gene mutation is seen in a variety of human cancers like retinoblastoma, osteosarcoma, neuroblastoma and so many other human cancer. P53 gene, or also known as TP53 gene, this is also known as guardian of the genome, located at the locus of short arm of chromosome 17, locus 13.1. Now we will discuss about how P53 works. When an injurious agent like ionizing radiation, carcinogen and some mutagen, when they expose to a normal cell in which P53 is in normal condition, there will be a DNA damage of this normal cell. This DNA damage will result into the accumulation of P53 which bind to the DNA, or some oncogenic stress and hypoxia may also result into the P53 gene accumulation and it binds to the DNA. Now these damaged DNA cell can have four future. They may go to the senescence, or these cell may activate P21, which is a cyclin dependent kinase inhibitor, and arrest cell in G1 cell cycle stage, or activation of growth arrest and death domain 45 factor, which is a DNA which play an important role in repair of the DNA. If the repair is successful, then there will be a normal cell in cell pool, or if the repair fail, this may stimulate the apoptosis and result into the cell death. And the last fourth mechanism is P53 activate pro-apoptotic factors like BEX and PUMA and will result into the apoptosis of the damaged cell. So we will discuss it again. A normal cell after exposure to the injurious agent will result into the DNA damage. This result in accumulation of P53 gene. Now the fate of this cell can be either senescence or activation of P21 and GADD45 will result into the successful repair, and cell may go to the normal cell pool, or if in case the repair fail, then they will stimulate the various mechanism of apoptosis and result in the cell apoptosis and cell being eliminated. But in cells, on the other hand, cells with the mutated P53, if such cells exposed to ionizing radiation, carcinogen and mutagen, they do not have the normal functioning P53. So these cells accumulate the DNA damage. P53 dependent genes not activated because of the mutant P53, and mutant cell, there will be the accumulation of damaged DNA, which is on additional mutation transform into the malignant tumor. So that is how a P53 gene prevent a normal cell from developing a cancer by these four mechanisms: either senescence, cell repair, or promotion of the apoptosis. Thank you.